Hello, I'm Brian Charlesworth. That's me in the left-hand corner. In the right-hand corner is Fred the cat, who's been sitting on my lap while I've been preparing this talk. I'd first like to thank SMBE for the Motoo Kimura Award. It's a terrific honour to have received this award, and I'm really grateful to the Society. I'd like to talk to you today about the properties of low recombination regions of genomes. This is something my group's been working on for quite a few years. Why should one be interested in this subject? Well, I think a lot of people are interested in the evolution of sex, and knowing what goes on when there's little or no genetic recombination should shed light on what goes on when there's little or no sexual reproduction. Furthermore, we know quite a lot from population genomic and molecular evolution studies about the properties of these regions, and that's what I'll tell you about today. First, it's been known for a long time that regions of the genome with low rates of genetic recombination often show reduced levels of DNA sequence variation. More generally, the level of silent nucleotide site diversity in a given bit of the genome is correlated with its local rate of recombination. There's a good deal of evidence, which I don't have time to go into, that this correlation is caused by increased effects of genetic hitchhiking when recombination is infrequent. Genetic hitchhiking, as I'm sure you all know, is what happens when selection at one place in the genome alters allele frequencies at nearby sites, even if they themselves are not subject to selection. Hitchhiking can involve both selective sweeps of favourable mutations spreading through the population and the elimination of low-frequency deleterious mutations, background selection, or BGS. Hitchhiking can also cause an increased skew of the site frequency spectrum, that's the distribution of frequencies of variance in populations, towards an excess of rare variance compared with neutral expectation, especially when there's little recombination. In addition, in Drosophila, regions of the genome that lack crossing over often show little evidence for adaptive evolution of protein sequences in contrast to what you see with normal levels of recombination, and codon usage bias is also reduced in these regions. This reduced level of adaptive evolution probably reflects the fact that there's more intense Hill-Robertson interference when you don't have much recombination. Hill-Robertson interference is what happens when selection at one site in the genome reduces the efficiency of selection at nearby sites. Here are some results from our own studies of populations of Drosophila melanogaster and a comparison with a closely related species. What we've done is to take groups of genes and bin them according to their rates of crossing over. That's on the x-axis, centimorgans per megabase. For each group of genes, we've estimated two parameters of the rate of adaptive evolution of the protein sequences controlled by the genes. Alpha in the top panel is the proportion of amino acid substitutions between the two species that are estimated to have been fixed by positive selection. Omega A in the bottom panel is the rate of evolution caused by positive selection relative to the neutral expectation. The green is for the autosomes, the red is for the X chromosome, the open circles are for genes with regions of crossing over, the filled circles for genes in regions where there's no crossing over but there's probably gene conversion going on. As you can see, there's little or no evidence for adaptive evolution of protein sequences in the non-crossover regions. This lack of evidence for adaptive evolution in non-crossover regions suggests that background selection may be the most important factor operating in these regions. But interpreting their properties is difficult because there's likely to be Hill-Robertson interference between the mutations causing background selection. This interference weakens selection against the mutations, causing them to experience drift around their equilibrium frequencies, which is much less marked when crossing over is frequent, and then you can assume mutation selection balance, which makes the predictions much simpler. Theoretical models of the effects of background selection in the absence of crossing over show that the interference effects cause much smaller reductions in variability than you get with classical background selection, but reductions which are consistent with the observed levels of variation in non-crossover regions of the fly genome. In addition, the models predict greater distortions of the site frequency spectrum towards rare variants when crossover frequencies are low. But observations on populations of both Drosophila and humans fail to show less skew with more crossing over. Here's what we see in Drosophila simians, a recent study. Two populations, Madagascar and Kenya. The top two panels show the diversity at fourfold sites as measured by pi, the diversity between pairs of alleles. And as you can see, this diversity goes up with the rate of crossing over. We bin genes in the same way as with the melanogaster study and express the rate of crossing over compared with the genome-wide average. The bottom panel shows a measure of skew, delta theta W. That's the difference between Waterson's theta and pi normalized by Waterson's theta. Waterson's theta simply counts numbers of sites which are segregating and is well known to be larger than pi when there's a skew towards low frequency variance. So a larger value of delta theta W tells us that there's more skew towards low frequency variance. And as you can see, there's little evidence for increase in skew with lower rates of crossing over. Indeed, the lowest crossover regions of the genome seem to have, at least for the autosomes, the lowest levels of skew. This is an inverse measure of skew estimated by Fanny Pouillet in Laurent Escoffier's group, graphed against the rate of recombination for several human populations. So the bigger this statistic, the less skew. As you can see, it starts off going down as the crossover rate goes up, so there's an increasing skew as the crossover rate goes up, and then it turns over and the skew goes down with increased crossing over. So there's a non-monotonic relationship between skew and rate of crossing over in humans. So we've got a paradox here. And indeed, simulations of models of the joint effects of background selection and selective sweeps with parameters that we think are realistic for Drosophila predicted a mean delta theta W of about 0.25. I'll show you some of these results later. However, a meta-analysis of 10 independent non-crossover autosomal regions from several species gave a delta theta W on average of about 0.18, which is quite significantly different. So we've got less skew than we might expect. And in fact, given that population expansions contribute to skew in many species, and this causes an increased skew towards low frequency variance, we might even expect much bigger skews than these theoretical predictions. So how do we resolve this paradox? Well, we noticed that our standard simulations assumed that deleterious alleles causing background selection were semi-dominant. In other words, the fitness of heterozygotes is reduced by half the selection coefficient against homozygotes. We also assumed a wide distribution of selection coefficients, in fact a gamma distribution with a shape parameter of 0.3. So this means there's a substantial fraction of deleterious mutations where the scale selection coefficient, 2NES, is around 5 or less, as well as a large fraction with S much bigger than 10. That's important. This suggested the possibility that if we change to a more realistic model of partially recessive deleterious mutations, a significant proportion of mutations might be experiencing what's called associative overdominance, or AOD. This is a process by which genetic drift generates linkage disequilibrium between neutral sites and linked sites subject to mutation to deleterious alleles. The LD can cause the alleles at the neutral loci to behave as though they're under selection favouring heterozygotes, because homozygosity at these sites is associated with homozygosity for somewhat recessive deleterious mutations. The loss of variability by drift at a neutral locus is then opposed by selection rather than accelerated as with background selection, despite the fact that both processes involve selection against deleterious mutations. AOD was studied quite intensively at the end of the 60s and the early 70s, but it's been rather forgotten about since then. So I had a nice interaction with a visiting graduate student from Shanghai, Lei Zhao, who did a very thorough theoretical analysis of background selection and AOD for just the simple case of one selected locus with deleterious mutations and another neutral locus. What we found was that when linkage is tight, so the scaled recombination rate 2NER is order less than 1, you can get AOD effects. You also need the mutations to be somewhat recessive, dominance coefficient less than a half. And finally, the scale selection coefficient, 2NES, must be quite small, of the order of 2.5 or less. This means that when selection coefficients are much bigger than this, you always get background selection, you never get AOD. In addition, because AOD causes neutral sites to behave as though they're subject to balancing selection, it should increase the lengths of external branches of the coalescent trees. This should cause site frequency spectra to be distorted towards a deficiency of low-frequency derived variance over neutral expectation, whereas background selection in low recombination regions has the opposite effect of causing an excess of low-frequency variance. With the extreme reduction in variation in non-crossover regions, it's clear that NE is going to be much reduced, so that 2NER must be much smaller than in crossover regions. If there's a wide distribution of selection coefficients, the more weakly selected deleterious mutations may experience NES values within a range compatible with AOD rather than background selection, although BGS may be operating at the more strongly selected loci in the region. AOD and BGS may thus be working against each other, so that the net skew in the site frequency spectrum towards rare variance is somewhat reduced compared with what happens with pure background selection. These expectations were confirmed by computer simulations of pieces of the Drosophila genome that were carried out by my former postdoc, Hannes Becker. We ran combinations of selection and recombination parameters that we thought were realistic for Drosophila. On the x-axis, you can see the rate of crossing over expressed relative to the genome-wide average, going all the way from no crossing over to the genome-wide average. The top two panels show the neutral diversity at sites embedded in the selected sites. The bottom two panels show our measure of skew, delta theta W. The left-hand side are results when there's no dominance, H equals 0.5. The right-hand side is when H equals 0.2, partial recessivity. The orange dots and arrows are for selection against deleterious mutations alone. And if you look at the arrows, these point to the cases when there is no crossing over, and you can see the skew is very much less when there's H equals 0.2 than when H equals 0.5. And you see the same with the purple dots and arrows, which is when there's a combination of background selection and selective sweeps. But the turquoise dots show no such pattern. This confirms that when you have partially recessive mutations, you can have a mixture of associative overdominance and background selection, reducing the skew compared with what you get with background selection alone. So, what are the conclusions here? Well, low recombination regions of genomes showed low levels of genetic variability and low rates of adaptive evolution. There's strong evidence that these properties are caused by stronger hitchhiking and Hill-Robertson interference effects in such regions. And selection against deleterious mutations probably plays a major causal role in the patterns that we see. However, the expected pattern of an increased skew towards rare variants when you've got low recombination rates is not seen. And this is likely to be due to associative overdominance caused by weakly selected, partially recessive deleterious mutations. And this finding adds a new layer of complexity to our understanding of patterns of molecular variation across genomes, which needs to be taken into account in future investigations. There are, of course, some problems. One question is why the Drosophila data don't seem to show the simulated pattern of a steady reduction in skew with increasing crossing over rate. In addition, we see different patterns in different populations and different species of Drosophila. And also the human data don't show a monotonic relationship between skew. We see a reduction in skew at very low crossover rates, followed by an increase, and then a steady reduction as the crossover rate increases again. A probable answer to these paradoxes is that the theoretical models have assumed constant population size, whereas natural populations have histories of population size change. Different regions of the genome have different effective population sizes due to the effects of selection at linked sites, and it's well known that different effective population sizes causes a different rate of response to past changes in actual population size. So it's a challenge to theorists to integrate models of selection at linked sites with demographic factors such as population size changes in a way that we can explain the complex patterns that we see in genomes in real populations. And I hope to live long enough to see progress towards resolution of this paradox. I'd like to thank Hannes Becker, Jose Campos, Penny Handrell, Dan Halligan, Ben Jackson, Vera Kaiser and Lei Shah for contributing to the work that I've described here. They and many other lab members over the years have been a wonderful bunch of people to work with, and it's been a real privilege to associate with them. I'd also like to thank the chief funders, BBSRC, the China Scholarship Research Council, the School of Biological Sciences of Edinburgh University, the Leverhulme Trust and the Royal Society. And I'd like to thank you for listening to me.